Greetings and welcome to Plus Therapeutics. It is now my pleasure to introduce your host, Mark Hedrick, President and CEO of Plus Therapeutics. Thank you, Mr. Hedrick. You may begin.
Thank you, Laura. Good afternoon, everyone. As Laura said, my name is Mark Hedrick. I'm the President and CEO of Plus Therapeutics. I'm pleased to be with you virtually today and hope you and your families and businesses are doing well. I'm very excited to present the latest on Plus Therapeutics to you. Our goal at Plus is to create innovative cancer drugs using nanoparticle drug encapsulation technology of both cytotoxic and radionuclide compounds. Our sweet spot is in the development of treatments for rare cancers that are poorly addressed or underserved, but represent an annual market potential in excess of $250 million per year. We focus our efforts on drugs that combine known APIs that are married to novel formulation or delivery approaches. We believe our development formula lowers risk without compromising upside opportunity. We pride ourselves on knowing both the technology landscape and the partner universe while having the core competencies to bring drugs from late preclinical to approval. Just recently, we announced the in-license of a very exciting portfolio of nanoparticle encapsulated oncology drugs for a variety of cancers. The lead candidate is currently in a phase 1 safety trial for glioblastoma and fits perfectly within our development model that I just mentioned. The technology was developed by a highly regarded group of U.S. cancer centers led by neuro-oncologist Dr. Andrew Brenner. The deal economics are heavily success-weighted, and the current phase 1 and planned follow-on trial have committed funding by the NIH and National Cancer Institutes. This recent transaction, when married to our existing technology and capabilities in nanoparticle drug encapsulation and development, position us to take a leadership role in two sizable market opportunities and bring forward a broad range of proprietary drug candidates. Nanoparticle liposomes are biologic envelopes that we make and we can customize. On one hand, we can change the delivered active agents in the payload bay. And we can also change the tissue selectivity and targeting based on what's engineered on the outside of the envelope. It's just a highly versatile platform. To make it all happen, I'd like to highlight two keys to our success. First, we have a small, exceptionally talented, full-time executive team supported with accomplished consultants that in total have decades of relevant, successful drug development experience. Second, we have a fully capitalized 10,000 square foot R&D and manufacturing facility in Texas. The facility is capable of commercial scale manufacturing of complex cytotoxics and radioactive compounds. With the recent deal, our pipeline continues to expand, now with our newest assets, the RNL product candidates. The lead RNL clinical indication is for recurrent glioblastoma. Additionally, there are a number of other targets in preclinical development. We also have two other assets in clinical phase, DocePLUS for small cell lung cancer and DocePLUS for 4 cancers, including breast and ovarian cancer. Given the recent deal, I'd like to focus the presentation on our newest assets, the RNL or radionuclide liposomal platform. First of all, let me give you a little background. Medical radionuclides represent a diverse group of medical compounds using a number of radioactive isotopes and represent a diverse group of diagnostics, therapeutics, and in some cases they have mixed use, which are also called theragnostics. The aggregate market for these compounds is anticipated to continue its steady double-digit growth well into this decade. In short, we see some great opportunities ahead for us. Furthermore, attractive deals continue to happen in this space for promising new drugs for cancer therapy. New FDA approvals have been seen in both rare cancers and those that are more common, and the drug pricing for these recent FDA approvals represent attractive economic opportunities based on the annual pricing for these drugs. Our lead RNL indication uses Rhenium as the active radionucleotide. Isotopic Rhenium is not easy to come by. It's actually made in a nuclear reactor, but it has some very unique properties. Importantly, it emits two kinds of radiation: beta energy for cancer killing and gamma energy for imaging. The imaging component is quite unique and important because it facilitates real-time visualization of the therapeutic at any point in time following treatment. So despite these unique properties, the use of Rhenium fits seamlessly into the typical hospital nuclear medicine workflow and is already approved now in Europe for palliative care for bone metastases. So now let me walk you through the therapeutic construct itself for our lead RNL drug. And there are actually four key components to the therapy: the Rhenium, the proprietary chelation technology, the liposome, and critically the means of delivery. As for the Rhenium, as I mentioned, it's a dual emitter, both therapeutic and imaging. It's an ideal isotope based on the property that it can distribute radiation 2 to 4 millimeters beyond the actual end of the atom and sticks around for a number of days. It has a 90-hour half-life. The proprietary BMEDA isotopic chelator is required to form a stable nanoliposome with Rhenium or other isotopes, and is a key part of the overall mix. The nanoliposome or envelope is a nanoscale construct of approximately 100 nanometers in diameter and facilitates the delivery of several hundred gray to the tumor. And then finally, delivery. We use convection-enhanced delivery that uses both hydrostatic pressure and time to fully distribute the active pharmaceutical ingredients, allowing coverage of the entire tumor bed and local tumor infiltration. So that's the therapy. Now about the indication, glioblastoma. Glioblastoma has been a really tough nut to crack. Although it's classified as a rare cancer, it's common enough that most folks know somebody that's had it. It's locally aggressive and it has a very poor prognosis. Patients succumb to the local spread, not actually the distant metastases. So from a therapeutic perspective, local control is the key. For a patient that is diagnosed and receives primary treatment, tumor recurrence is the norm. And when it does recur, the standard of care is far from well defined. There are several options, but none work particularly well, and there's only been one approved new therapy since 2011. External beam radiation therapy, or EBRT, is the best known killer of glioblastoma cancer cells and is the mainstay of primary therapy, but it's limited by toxicity to an overall dose of about 80 gray. So clearly some better solutions are needed here for these patients, and it's a very tough diagnosis. RNL for recurrent glioblastoma has many theoretical advantages over standard of care in EBRT. Here are 5 key ones that come to mind: retention, targeting, tumor coverage, delivery, and dosing. Retention such that the therapy stays at the tumor and has a long half-life. Targeting in the sense that the radiation can be taken up by tumor glial cells, and the imaging component where the photons provide localization in real time. Tumor coverage so that radiation reach is expanded beyond the reach of any particular atom by 2 to 4 millimeters. Delivery such that using image-guided convection, enhanced delivery, and stereotactic delivery of a cannula to the brain solves the issue of how to get across the blood-brain barrier. And then finally, dosing such that at the end of the day, one may deliver 10 or up to 20 times the dose of radiation versus EBRT. So how does RNL generally stack up to EBRT? For primary glioblastoma, radiation is perhaps the most effective cancer killer, and the survival contribution to combined therapy is considered to be about 5 months. Now, there should be no ambiguity here on the mechanism of action. Radiation kills cancer cells very well. But for EBRT, the radiation must pass from outside in, if you will, meaning it must go through normal tissues to get to a tumor, hence the strict limitations on delivery. RNL, in contrast, is delivered inside out at much higher dosages, residing locally until its radiation energy is dissipated. Thus far in our studies, no serious adverse events have been observed despite the multifold increase in radiation. In comparison with EBRT, and now we're talking about in the recurrent setting, just look at the numbers. There's really no contest in terms of how much radiation energy can be delivered. And for RNL, it's a single shot versus for EBRT, that's 5 times a week over 4 weeks. And also, I think there are a number of other potential benefits as well. So this next slide is really a key slide in terms of understanding why this technology has advantages over other approaches. The nanoparticle construct that Dr. Brenner and his colleagues developed provides excellent retention and tumor coverage or dispersion. This RNL construct, which as I mentioned is a liposome, stays around the tumor for days, given the long half-life, and can cover the tumor in its entirety. In the graph above to the right, you can see other formulations using the same radioisotopes don't stick around in the brain or in the tumor and exit the scene rapidly. It's time on tumor that's critical. Prior to commencing the phase 1 trial, 2 relevant preclinical studies of RNL showed no safety issues and promising survival signals in treated subjects over control, in this case rodents. It's really amazing. These animals tolerated about 30 times, that's 3,000% more radiation that would have been given with the next best option, EBRT. In the same study, looking at the MRIs of control animals, they show the typical rapid tumor size increase and progression at 2 weeks. That's on the bottom of the slide. At the top, while the treated subjects, the tumors regressed over 70 days post a single administration. The IND-enabling toxicology studies confirmed the ability to administer big doses of radiation with, in this case, up to 360 gray with no adverse effect limit on brain pathology. In other words, lots of radiation can be given without hurting normal brain tissue. So what's the current status of this program? RNL is in a multi-center volume and dose finding study of the safety, tolerability, and distribution of RNL directly administered by convection enhanced delivery to patients with recurrent or progressive malignant glioma after standard surgical radiation and or chemotherapy treatment. So today, 13 patients with recurrent glioblastoma have been enrolled, and we are currently enrolling in cohort dose 5. Absorbed doses are now over 500 gray that have been delivered. That's about 10 to 15x what is typically delivered by EBRT. There have been no treatment-related adverse events higher than grade 1. And their early efficacy signals, the survival in the first 2 patients was greater than 2 years. Our goal is to complete enrollment in 2020. And as I alluded to earlier, this trial and its subsequent phase 2 are supported by an NIH and NCI grant. So the therapeutic that we're putting into these patients is complex scientifically, no question there. But I think it's helpful to look at it from a patient's perspective. For them, it's actually a pretty straightforward therapy. And let me explain to you what happens. At any time prior to admission, specialized software plus the MRI and CT scan information are used to calculate ideal catheter trajectory, catheter number, and depth. And this is to avoid critical brain structures, but also help fully cover the tumor with radiation. The software also uses complex algorithms to help predict a priori the distribution volume of RNL to the tumor over time. On the day of admission, the patient goes directly up to the OR and using geospatial positioning and stereotactic guidance based on the coordinates determined in the preoperative in silico surgical planning process, 1 or up to 4 catheters are inserted into the tumor, whatever is required for that particular patient. The patient then remains in the hospital for observation and to ensure proper catheter positioning prior to treatment. On the following day, the RNL is administered by convection-enhanced infusion over several hours. SPECT and CT scanning is used in real time to confirm the actual dispersion and localization of the RNL. After the infusion is concluded, the catheter is then removed, and the following day, presumably with no problems, the patient's discharged. And then the CT SPECT is used to follow up the post-infusion distribution volume and location. And you can see the CT SPECT data on the slide at 40 minutes during infusion, going through 24 hours, and then at 5 days after infusion. As I mentioned, it's too early to draw any meaningful efficacy conclusions, but here's some of the key clinical details from the first patient, patient 001. In this patient, there was no clearance of radioactivity during the initial 72 hours. Local regional retention was over 95% of the administered radioactivity from time 0 to 72 hours. The average radiation absorbed dose to the 4.3 mil of tumor volume was 143 gray, again, a very big dose. Over 80% of the tumor volume was effectively covered by the radiation, at least out to 5 days, and in terms of outcome, this patient survived for 30 months. So what about next steps? For the phase 1 trial, we plan completion of enrollment in late 2020, assuming minimal COVID-19 impact. In fact, we continue to screen patients, and we're working to add 2 new sites. We hope to have manufacturing, supply chain, and product scalability in place by mid-2021. In parallel, we seek U.S. and EU regulatory feedback and consider options such as orphan designation, possible accelerated paths later this year. Simultaneously, we'll prepare for a phase 2 pivotal trial with the hope to initiate in second half of 2021. Switching gears, as part of the RNL transaction, we obtain rights to other indications in RNL formulations, and I'd like to highlight 2 of those. Abdominal or peritoneal cancer spread, called carcinomatosis, occurs in several abdominal tumor types, such as with ovarian, colon, and stomach cancer. Like glioblastoma, there are no good treatments. In preclinical studies, direct abdominal delivery of RNL is showing promise in reducing tumor burden and improving abdominal swelling and carcinomatosis. And this micro PET scan, purple, the color purple is bad, and the color green is good. Treated animals at 4 weeks showed a gradual decrease in signal or tumor versus the control group that showed intense peritoneal uptake indicating tumor growth. Compare the bottom center panels on each the treatment and control group. In a different cancer, squamous cell carcinoma, typical in the head and neck, RNL shows a similar pattern of tumor killing with direct tumor injection as would be seen in recurrences in patients. We are evaluating these additional development opportunities plus others in the Q2 timeframe, and there's more to come here over the next few months. As I mentioned earlier, we have 2 additional nanoparticle drugs with cytotoxic payloads. DocePLUS is a liposomal formulation of docetaxel, the API found in Taxotere. The goal of this formulation is to improve tumor killing capability that has better pharmacokinetics than other formulations of docetaxel. DocePLUS has a better safety versus efficacy trade-off in a number of preclinical tumor models. And in the phase 1 trial, DocePLUS achieved a 20% increase in the maximum tolerated dose, improved pharmacokinetics, and promising efficacy signals in a heavily pre-treated patient population. In fact, a third of these patients had prior Taxotere use. The best tumor responses, measured by a decrease in tumor size, seem to be both in lung and pancreatic cancers. As for next steps for the DocePLUS and Doxo programs, we are reevaluating these in light of our recent RNL acquisition and holding off today on any further development spend pending partnerships, which we are currently seeking, and I'll have more detailed plans on this later in Q2. Now, onto our capitalization table. In 2019, we raised approximately $28 million in a series of transactions and divestitures. We finished the year with $17.5 million in cash and recently restructured our senior term loan such that it now matures in 2024 and has a current principal balance of $4.3 million. Also last year, the company, through divestitures and restructuring activities, substantially reduced its cash burn and relocated its headquarters to Austin, Texas. Now finally, as to milestones. In 2019, you may recall we guided to an expanded pipeline and restructured term loan. Both now have been announced. Going forward in 2020, we plan to close out the RNL in-licensing transaction soon, optimize the regulatory and clinical program for RNL and glioblastoma, and get FDA feedback on the next steps. We also hope to complete the enrollment on the phase 1 trial and report data from the phase 1 and nail down the phase 2 pivotal trial plan. We also plan to expand the pipeline in part through follow-on RNL indications, and we continue to be disciplined but aggressive in looking for acquisition or in-licensing of new pipeline candidates. And finally, we continue to seek out-licensing and partnering opportunities for DocePLUS, DocePLUS, and now our new RNL assets. So with that, I want to thank you very much and be well. Appreciate your time.
This concludes today's webcast. You may disconnect at this time. Thank you for your participation.